So another key component we have to consider in this pharmacokinetic phase, that is the plasma protein binding. So plasma protein binding, like we know that if our drug is highly lipid soluble, then the plasma protein binding will be high. But high, that means not that very high is not desirable for anything. It's like a general rules of thumb. So excess or too much low is not that good. So if generally we see that our drug most of the time has a above 90% of this plasma protein binding. So if we see that it is like more than 99%, then it is a sign for danger. So like most of the drugs will not be available for its function, for its pharmacological action, because I know that only free drugs. So this is in this free state. When the drug is free, this is available for the action. But when the drug is bound to the plasma protein in the blood, then the drug is not free, or the drug is bound to the plasma protein and it is not available for its function. So this is one important factor, plasma protein binding. And like if we see this 50 to 90%, this is actually good. But if we see this is below 50%, then it is very good actually. So we don't have to consider anything about the plasma protein binding. But if we see high 99%, then for a toxic drug that has a very narrow therapeutic index, that then it will be very harmful or very dangerous for those drugs, high potent drugs. For instance, the cardiac drugs, especially the digitally, so those are in dose like microgram. So microgram are therapeutic dose, and like the safe zone is very narrow, so you cannot like those dose calculations are very strict, and those drugs cannot be allowed to bind too much to the plasma protein binding, or other drugs, those mainly that displace the plasma protein binding to other drugs. So their drug interaction profile is very strict and very dangerous, because toxic drugs may highly bound to the plasma protein, but if we take another drug that also have high affinity for the plasma protein, or mainly those high affinity drugs mainly displace other drugs from the plasma protein, then in that case the highly potent drugs will be more available in the blood, and this will cause some serious toxic effects. So highly plasma protein bound drugs with a narrow therapeutic index is actually harmful. So these are the mainly consideration of the plasma protein binding. So high binding is not that good. So moving to the next slide. So distribution, or mainly denoted by the volume of distribution. So it mainly indicates that when the drug is absorbed, when the drug has reached the blood, then the drug starts to distribute throughout the whole body, like the tissue compartment, fat compartment, etc. So if we see a high VD, volume of distribution, that means the drug is more accumulated in the tissue regions or the fat regions. So if we see the volume of distribution is low, that means the drug is in the aqueous phase or in the blood most of the time. Also, we have to consider the concept of bound drug or unbound drug. Unbound drugs are free, but bound drugs, like the bound to the plasma proteins, are not free. Those are not available. So the distribution mainly refers to the bound. Also, the distribution also depends on the bound and unbound state of the drugs. So if our drug is at the unbound or free state and it is in the plasma, then we can say safely that the drug will reach our site of action in a sufficient amount. But if the drug is bound state or accumulated in the fat tissues or stored inside the tissue because of its high lipophilicity, then we cannot say this drug will reach our target of tissue at a sufficient time or at a proper time. So the pharmacokinetic profile of the drug will be unpredictable and could create problems at the later when the drug redistributes in the blood, causing the plasma concentration high or fluctuate. So distribution is another important parameter to consider. So going to the next slide. So metabolism. So like all other parameters, metabolism is another crucial parameter because the rate of metabolism mainly determines the required dose, or the potency, or the overall profile of the drug. So metabolism is very important. So we know that metabolism is composed of two parts mostly, that is biotransformation and excretion. In total, they are called metabolism. So biotransformation mainly happens in the liver largely, and excretion mainly largely occurs through the kidneys. We know that. So the purpose of the metabolism or biotransformation is that the drugs are lipid soluble, right? So for the drug to excrete, the drug needs to be water soluble. So the lipid soluble drugs undergo conversion, enzymatic conversion, in order to make it more water soluble, so that it can easily excrete through the kidneys or the water phase. So in the kidney, so aqueous phase drugs or highly water soluble drugs are excreted through the kidneys. But if the drug is lipid soluble, then it needs to be converted further. So we know that largely there are conversions are divided into two phases. The reactions are divided into two phases, phase 1 and phase 2. So, and there is another phase 3. So that is not shown, but the transporters, the efflux transporters in the liver or kidneys, those mainly pump back the drugs in the blood. So those mainly referred to as the phase 3 conversions. So for now, for the simplicity, phase 1. So phase 1 are very basic reactions like the oxidation, reduction, hydrolysis. Those are the phase 1 reactions. And phase 2 are the conjugated reactions like addition of the adducts, addition of different conjugates throughout through the compound like glucuronic acid or sulfonic acid. So those are the conjugation or phase 2 reactions. But most of the time the drug undergoes both of the phases. At first the phase 1 reactions, and after that the drug undergoes phase 2 reactions in order to make it more water soluble, so that it can excrete through the kidneys. So just note that one important enzyme family in our body, that is the cytochrome P450 or CYP450. So these are the large family of enzymes that mainly converts or metabolize the xenobiotics. Xenobiotic means the drug or any foreign particle, foreign molecule in our body. So another isoform like CYP450 is a large family of proteins, so it has a lot of isoforms. The isoform CYP3A4, particularly this, mainly metabolize important drugs. So note the names 3A4, 2AB, different CYP enzymes. There are more than 1000 isoforms of cytochrome P450. So from cytochrome, that means the drugs are converted. Also this called the microsomal conversion. If you can recall the metabolism, the microsomal and non-microsomal. So microsomal conversions of the metabolism of the drug mainly done by the cytochrome, like those conversion in the endoplasmic reticulum forming the microsome, or in the mitochondria, those conversions happening inside the cell. So we will see that how can we measure in vitro this metabolism by taking up the liver tissue or the microsomal or endoplasmic reticulum, by isolating the endoplasmic reticulum from the liver tissue. So another thing is the polar molecules. We know that polar molecules are water soluble, and non-polar molecules are lipid soluble. So if our drug is more lipid soluble, then the conversion makes it more, or the metabolism makes it more polar molecule, so that it can be dissolved in the water. So that's important point. So metabolism, we know that. So these are the basic steps. Like the prodrug needs to undergo metabolism first in order to carry out its action. So this enzyme P450, cytochrome P450, very important enzyme of family for drug metabolism. So at therapeutic concentration of most drugs, metabolism rate increases with high concentration. It is known that we all know that if the concentration of the drug is increased in the blood, that means the drug will more permeate, or more perfusion will increase through the metabolic organs such as liver, so that the metabolism rate will also increase at the same time. So going to the next slide. So how can we measure metabolism before, before testing in the human, or before testing the mouse animal? We can do that in vitro, like we can isolate liver cells. Like there are two ways actually. The microsomes. So I mentioned that what are the microsomes? Like the microsomes are mainly the endoplasmic reticulums. So in the endoplasmic reticulums are liver enzymes. In the liver cells of the endoplasmic reticulum, the enzymes mainly reside. So two ways, like the microsomal assays and hepatocyte assays. So in the microsomal assays, we take liver cells and fraction it and centrifuge it to get the microsomal part. Microsomal part is specific to the conversion, and then we test the compounds, and see the rate of metabolism. And the other way, this is more like relevant, the hepatocyte assays. We take the whole liver cells and we test our compounds in the whole liver cells, whereas in the microsomal assays we test the liver cells endoplasmic reticulum fraction, because endoplasmic reticulum, mainly cytochrome P450, metabolic conversion of the drugs happens in the endoplasmic reticulum, and to some extent in the mitochondria. So after testing, we get an idea of the intrinsic clearance. So see clearance intrinsic, that how milliliter of the drug per minute is cleared from that liver. Then we use this data to predict the profile in the in vivo, and how they would behave in the in vivo. So also general rules of thumb is that increasing the lipophilicity generally increases the metabolism. So this mainly adds more point for the metabolic enzyme to work. So for instance, here is an example. So if this lead compound and this marked zone is the site of metabolism, that means the enzymes can catalyze this site. So if we can block this site, we have done some modifications, then we can potentially block this site from the enzymes action, metabolic enzymes action. So what can I do? Like if we replace it by the two fluoride ions, that means here you can see there are, in this carbon there are two hydrogens present, right, in this portion. So then by replacing those two hydrogens by two fluoride, we can block this site of metabolism. But note that this blocking, this fluoride, adding of the fluoride, it also increases the log D, that means the lipophilicity. So this addition or modification is good for this one site, but it also increases the metabolism. So it potentially will increase the other site of metabolism. So like here it can potentially opens the door for other enzymes to work. So it is not a safe modification. So what can we do? We can either block it. We must have to block this site, as well as we have to lower or at least maintain the lipophilicity. So by replacing it with the oxygen molecule. If you can see in the next modification here, it also by adding this, replacing those fluorides or replacing this carbon with this oxygen, totally blocked the metabolic site. Also it lowered the lipophilicity, which is good. So if we also modify to this with this group SO2, potentially this will also block the metabolic site, also um, lower the lipophilicity. So these two examples are good compared to this one. So this is one way of like manipulating or blocking the metabolic site from the enzymes. So if our compound has more than one sites, then we have to consider other metabolic sites too. So as well as we have to maintain a balance, like we are blocking one site, and is it causing or reducing or increasing the lipophilicity or not, we have to take account that too.